This is Adriana Galvan from the Department of Neurology at Emory University. This presentation is an overview of the chapter Anatomy and Synaptic Connectivity of the Basal Ganglia. In this presentation, I will briefly go over some of the principal concepts covered in more depth in the chapter. The basal ganglia are a group of interconnected subcortical structures. In these two corneal sections from a human brain, we can identify the basal ganglia. The dorsal striatum, comprised by the caudate and the putamen. The ventral striatum, also known as nucleus accumbens, not shown in this image. The external and internal segments of the globus pallidus. The subthalamic nucleus. The substantia nigra, including the pars reticulata and pars compacta. And the ventral tegmental area, not shown here. Let's talk about the connectivity of the basal ganglia. In this diagram, the blue box indicates the interconnected basal ganglia nuclei that receive extrinsic inputs from the cortical and thalamic regions. The striatum and the subthalamic nucleus are the main entry points for extrinsic glutamatergic inputs. Cortical glutamatergic inputs to the striatum originate from most cortical areas, while the thalamostriatal projections arise mainly from the caudal intralaminar thalamic nuclei, the centromedial and parafascicular nuclei. The basal ganglia output originates from the internal segment of the globus pallidus and the substantia nigra reticulata, and is directed towards frontal areas of the cortex via the thalamus and to other subcortical structures, including the lateral avenula and various brainstem structures. The basal ganglia are part of circuits that involve the cerebral cortex and thalamus. Depending on the function of the cortical regions from which they originate, there are three main functional segregated loops: sensory motor, limbic, and associative or cognitive. In each of these loops, specific functional cortical areas project in a topographic manner to specific areas of the striatum. This segregation of cortical information is maintained to the rest of the basal ganglia nuclei and also in the basal ganglia receiving regions of the thalamus. The majority of neurons in the striatum are spiny projection neurons. These cells are GABAergic. There is also a minor proportion of cholinergic and GABAergic interneurons. At the synaptic level, the major targets of corticostriatal terminals and a significant subset of thalamostriatal terminals are the dendritic spines of spiny projection neurons. The corticostriatal inputs also contact GABAergic interneurons. However, thalamic inputs from the CMPF contact mainly the dendritic shafts of spiny projection neurons and also striatal cholinergic interneurons, and to a lesser extent, the GABAergic interneurons. Striatal spiny projection neurons are divided into two groups based on their main projection targets and the type of dopamine receptor they express. The neurons projecting to the basal ganglia output nuclei, the GPI and the SNR, form the direct pathway since they reach the output nuclei of the basal ganglia directly. The neurons that project to the GPE form the indirect pathway since they reach the output of the basal ganglia through the GPE and the STN. Direct pathway neurons express preferentially D1 receptors, whereas indirect pathway neurons express D2 dopamine receptors. An imbalance of activity between these two pathways is a cardinal feature of the basal ganglia pathophysiology in Parkinson's disease and other movement disorders. The striatum receives a massive innervation from the ventral midbrain dopaminergic cells. This dopaminergic innervation plays a critical regulatory role over cortical and thalamic glutamatergic transmission upon spiny projection neurons through pre- and postsynaptic mechanisms. Dopamine neurons process and transmit to the striatum complex and heterogeneous information related to movement, reward-related stimuli, and aversive events. Along with the striatum, the subthalamic nucleus is considered an input nucleus of the basal ganglia. As for the corticostriatal system, the cortico-subthalamic projection arises from different motor, associative, and limbic cortical regions that innervate functional segregated regions of the STN. The STN also receives connections from the centromedian and parafascicular nuclei of the thalamus, the external globus pallidus, and the ventral pallidus, or VP. The subthalamic nucleus sends glutamatergic excitatory projections to the globus pallidus, the substantia nigra, the pedunculopontine nucleus, and the pontine nuclei, the latter being considered as a route through which basal ganglia information can reach the cerebellum. The internal segment of the globus pallidus and the substantia reticulata are the output structures of the basal ganglia. Both nuclei receive functionally segregated inputs from the striatum, and in turn send GABAergic projections to their thalamic and brainstem targeted structures. Through their thalamic projections to the ventral motor nuclei and the CMPF complex, cortical information is sent back to frontal cortical regions via segregated basal ganglia thalamocortical loops, or to specific striatal regions via basal ganglia thalamostriatal projections. The brainstem targets of the basal ganglia output nuclei include the superior colliculus, pedunculopontine nucleus, the lateral alveola, and the parvicellular reticular formation. Through its connections with most basal ganglia nuclei, the thalamus, and various lower brainstem structures, the pedunculopontine nucleus is in a strategic location to mediate widespread basal ganglia influence in the central nervous system while bypassing the conventional thalamocortical system. The lateral alveola is considered as an interface between the limbic system and the basal ganglia because it receives reward-related information from the GPI. The projections to the superior colliculus is important in the control of saccadic eye movements and visual orientation. Disruption of communication by degenerative neuronal loss or neurochemical changes in various nodes of the basal ganglia thalamocortical loops lead to the development of motor and non-motor signs of a large number of neurological and psychiatric disorders, including Parkinson's disease, Huntington's chorea, Tourette syndrome, dystonia, attention deficit disorders, and obsessive-compulsive disorder. In this figure, the left panel indicates the circuits in the normal state, and the right shows the overall changes in activity that had been associated with Parkinsonism. Green and red arrows indicate excitatory and inhibitory connections respectively. The thickness of the arrows corresponds to the presumed activity. As can be seen, the loss of dopamine innervation to the striatum after the generation of dopaminergic cells in the substantia nigra compacta triggers a series of functional changes in activity along the basal ganglia circuits, resulting in presumed increased inhibition from the output nuclei of the basal ganglia and reduced activity in thalamic and other targets. This functional model of the basal ganglia in normal and pathological states has guided research in the field for more than 30 years. Thank you for your attention. I invite you to read the full chapter and included references.